And dear students of 12th medical, today in botany we are going to take up the post-fertilization events. Structures and events rather. Previously we have been discussing the pre-fertilization events and we have reached up to the fertilization stage wherein we discussed the double fertilization. And now we will move ahead after the double fertilization. The events like the formation of the endosperm and embryo development take place in the ovules, and maturation of the ovules into the seeds and ovary into the fruit, and these are the post-fertilization events we are going to take up. Firstly, we will take up the endosperm. This endosperm is the food-laden tissue which is meant for nourishing the embryo in the seed plants. The primary endosperm cell divides repeatedly and forms a triploid endosperm tissue. Angiospermic endosperm is of three main types. That means the endosperm development takes place. It is of three types. First one is the nuclear endosperm, the cellular endosperm, and the halobial endosperm. Now the nuclear endosperm. The primary endosperm nucleus divides repeatedly without valve formation to produce a large number of free nuclei. Meanwhile, central vacuole appears in the cell and pushes the cytoplasm containing the nuclei to the periphery. The cytoplasm thickens so that the vacuole decreases in size. It ultimately disappears. The vacuole ultimately disappears. The multinucleated cytoplasm undergoes cleavage, gives rise to multicellular endosperm tissue. For example, in maize, wheat, rice, sunflower, the endosperm is multicellular endosperm. Whereas in a coconut, the coconut water from tender coconut is free nuclear endosperm with thousands of nuclei, and the surrounding kernel is cellular endosperm. And this is the view of the nuclear endosperm, wherein the free nuclei are formed in large numbers, like in the tender coconut water. And second one is the cellular endosperm. Every division of the primary endosperm nucleus is followed by cytokinesis, that is the valve formation, that is the formation of the cells. The endosperm becomes cellular from the very beginning, for example, balsam, datura, and petunia. This is the view of the cellular endosperm. And the third type of the endosperm is the halobial endosperm. It occurs in halobials. One of the orders in the plant kingdom is halobials, and the halobial endosperm is formed in halobials order. The endosperm is of intermediate type between nuclear endosperm and cellular endosperm. The first division of primary endosperm nucleus is followed by transverse cytokinesis to form two unequal cells, the micropylar cell, which is large in size, and the chalaza, the smaller sized cell. The micropylar grows fast, but the chalaza remains small and may degenerate. For example, in Asphodilus. Now the fate of the endosperm. This is the view of the halobial endosperm. The cell divides into two, the larger one the micropylar and the smaller one is the chalaza, which may later degenerate. And now the fate of the endosperm. The endosperm may be completely consumed by the growing embryo. For example, in pea, beans, before seed maturation, or it may persist in the mature seeds, as in castor and coconut, and it is used up during the seed germination. Thanks. Have a nice day.